Hey everyone, welcome back to School Subjects Online. Today we have an exciting list of biology experiments perfect for high school students. We'll be exploring DNA, cells, enzymes, bacteria, and even our own reflexes. So, put on your lab coats and let's get started. Have you ever wanted to see DNA with your own eyes? Well, now you can. We're going to use ordinary strawberries to do it. First, we'll mash up some strawberries to break down their cell walls. Next, we'll add a mixture of soap and salt to help release the DNA from the cells. Then we're going to filter the mixture through a coffee filter to remove all the big chunks of strawberry. When you carefully pour ice-cold rubbing alcohol into the strawberry liquid, watch closely. The DNA will start to come out of the solution. You'll see these white, stringy clumps, almost like cobwebs, forming in the alcohol layer. That's the strawberry DNA. Pretty cool, right? Mitosis is essential for life. You can see this process using onion root tips. First, we need to prepare the onion root tips by staining them so we can see the cells more clearly. Then we'll place the stained root tips on a microscope slide and have a look under a microscope. You'll see cells lined up in the middle, ready to divide, and others with their chromosomes pulled apart. It's like watching a choreographed dance of life unfolding right there on your slide. Photosynthesis is one of the most important processes on Earth. You can experiment with it using a simple setup. We'll start with a sprig of Elodea, an aquatic plant, submerged in water with a pinch of baking soda. We'll shine a light on the Elodea and observe what happens. As the plant absorbs the light and starts photosynthesizing, you'll see tiny bubbles forming on the leaves and rising to the surface of the water. These bubbles are oxygen, a byproduct of photosynthesis. This experiment shows us that light is essential for photosynthesis to occur. Enzymes are like tiny helpers in our bodies that speed up chemical reactions. One of the coolest enzymes is bromelain, found in pineapple. For this experiment, we'll use gelatin, which is made up of protein. We'll prepare 2 batches of gelatin, one with fresh pineapple juice and one with canned pineapple juice. The fresh pineapple juice will prevent the gelatin from setting because the bromelain breaks down the gelatin's protein structure. On the other hand, the gelatin mixed with canned pineapple juice will set normally. This is because the heat used during the canning process destroys the bromelain enzyme. Remember those helpful enzymes we talked about? Their activity can be affected by things like temperature and pH. In this experiment, we'll use liver and hydrogen peroxide to see how pH affects enzyme activity. Liver contains an enzyme called catalase that breaks down hydrogen peroxide into water and oxygen. When we add hydrogen peroxide to a piece of liver, we'll see bubbles forming. Now let's change the pH. Adding a little vinegar to the mix will make it more acidic. Observe what happens to the bubble production. Next, we'll add a pinch of baking soda to make the solution more alkaline. Watch the bubbles closely. By comparing the bubble production at different pH levels, we can see how pH affects the activity of the catalase enzyme. Bacteria are everywhere. In this experiment, we'll create a perfect environment for bacteria to grow and observe their amazing ability to multiply. First, we'll need some Petri dishes filled with agar, a nutrient-rich gel that bacteria love to eat. Using sterile cotton swabs, we'll collect bacteria samples from different surfaces, such as our phones, doorknobs, or even our mouths. Once we've inoculated our Petri dishes with the bacteria samples, we'll seal them tightly and store them in a warm, dark place. Over the next few days, we'll observe the dishes and watch in amazement as bacteria colonies start to grow. This experiment highlights the importance of hand washing and sanitation in preventing the spread of harmful bacteria. Ever wonder how fast your reflexes really are? This simple experiment will put them to the test. All you need is a ruler and a friend to help you. Have your friend hold the ruler vertically, with the 0 mark at the bottom. Place your hand just below the ruler, ready to catch it. Your friend will then release the ruler without warning, and your job is to catch it as quickly as possible. Measure the distance the ruler fell before you caught it. The shorter the distance, the faster your reaction time. To make it even more fun, try the experiment under different conditions. See if your reflexes are faster with background music, with distractions, or even after a cup of coffee. Blood is essential for life, carrying oxygen, nutrients, and fighting off infections. In this experiment, we'll create a model of blood using everyday household items. We'll start with a clear container to represent the blood plasma, the liquid part of blood. Then, we'll add red candies or beads to represent red blood cells, which carry oxygen throughout the body. Next, we'll use white beans or marshmallows to represent white blood cells, the body's defense system against infections. Finally, we'll sprinkle in some small dark sprinkles to represent platelets, which are responsible for blood clotting. Caffeine is a stimulant that can affect our heart rate. In this experiment, we'll observe the effects of caffeine on the heart rate of tiny aquatic creatures called Daphnia. We'll prepare different solutions of caffeine with varying concentrations. Then we'll carefully place a few Daphnia in each solution and observe their heart rate under a microscope. Daphnia are transparent, so we can actually see their tiny hearts beating. As we compare the heart rates of Daphnia in different caffeine concentrations, we can see how caffeine affects their heart rate. This experiment highlights the impact of stimulants on living organisms. Hand washing is one of the most effective ways to prevent the spread of germs. In this experiment, we'll see the difference hand washing makes in a visual and slightly gross way. We'll use slices of bread as our testing ground for germs. We'll have one slice that we'll touch with unwashed hands, another slice that we'll touch with washed hands, and a control slice that we won't touch at all. Then, we'll seal each slice of bread in a separate Ziploc bag and store them in a warm, dark place. Over the next few days, we'll observe the bread slices for signs of mold growth. This experiment clearly demonstrates the importance of hand washing in removing germs and preventing the spread of microorganisms. Wow, we've covered some seriously cool biology today. From extracting DNA to growing bacteria, these experiments are a blast to try and a great way to learn about the amazing world around us. Don't forget to like, share, and subscribe to School Subjects Online for more fun and unique ideas. Until next time, stay curious, keep experimenting, and always keep asking questions.